Yeah, all right, great. Yeah, welcome everybody. My name's Tom Schmidt. I'm a faculty member here at Michigan in the departments of internal medicine and ecology and evolutionary biology, and study the gut microbiome. It's my pleasure to introduce today's speaker, Dr. Patrick Chain. Patrick's a scientist at Los Alamos National Labs. He leads a group there that studies genomes and metagenomes from microorganisms, and has been hanging around Ann Arbor some recently because his wife is a resident in pathology. And so once I heard of that, I thought, well, great, this is a great opportunity to hear from Patrick. Patrick and I both worked on microbes and microbial communities in a number of environments. And the classic approach in microbiology, the way we learn, I think we still know the most about microbes by isolating them and growing them in the laboratory. That's how we study microbes. The problem is that lots of microbes that are in the environment, in the human gut or elsewhere, we haven't yet cultivated. It's my personal bias that it's because these microbes have formed networks with other microorganisms, and when we pull them out of that network, it's difficult to provide everything that they had been getting from other microbes in their environment. So one of the approaches that has been developed to circumvent that requirement for cultivation is just to reach into the environment, grab a sample, extract all of the nucleic acids there, and sequence them. So great, don't have to culture the organisms to learn something about them, but the challenge is now, what do you do? How do you interpret those terabytes of nucleic acid sequences that you get from the environment? So Patrick is one of the people who is really pushing the boundaries on how to do that. How do we make sense of these shotgun metagenomes and transcriptomes from the human gut and from other environments? So glad you're spending some time in Ann Arbor, and thanks for joining us today.
Thanks, Tom. Is this on? You can hear me? Yes? Good. So thanks for the invitation. It's a pleasure to be here, though I'm in Ann Arbor quite frequently these days, but still a pleasure to be on campus and talking a little bit of science with people that I can see not via video screen, which is where I conduct most of my work these days. I'd asked Tom what I should present or what flavor I should present, how much background, and he suggested I should just go with my gut. So you can blame him for anything that that you don't like about this. So yes, I know. Yeah, yes, that's what I was inferring. So for those of you unfamiliar with Los Alamos, I thought I'd give you a brief 2-second tour, since I was told by some individuals they weren't entirely sure of the environment. But it's located in this lovely place, which was, from what I understand, the only secret city in the US, and it was just a postal code in Santa Fe for quite some time before it became an actual city of Los Alamos. It's in northern New Mexico. We're at 7,000 feet elevation. We get nice snow. Well, sometimes we get nice snow in the winter. Ski Hill is about a 10-minute drive from my office. Elevation's about 10,000 feet at the top. But that's just the environment. This is the city. The county itself is only about 18,000 plus people. The lab itself employs almost 12,000, so you can imagine the types of discussions that go on in the fruit aisle at the grocery store. However, despite this large number, only about 200 people in Bioscience Division, which is where I'm located, and most of the people at LANL also ask me, why are you here and what do you do? Because it's a surprise to them that there's biology at Los Alamos. So this is the type of slide that I open up in any talk that I give at Los Alamos, is to try and poke the physicists and the modelers in terms of why genomics might be a type of science that the national lab should really invest in and embrace with open arms. So it's a big data science with a proposition to change the term astronomical to genomical, which I'm a proponent of. And the microbiomes won the cover, whereas supernova, machine brain, and fusion energy are just relegated to the top. So I present this normally as my first slide at Los Alamos. So this large data capacity, or this big data for genomics, is driven primarily by these new technologies. We were not stuck, but we were limited to one sequencing technology for about 20 years. And then all of a sudden, all these newer technologies came out with far higher throughput at a reduced cost. And this opened up a number of different avenues. It opened up avenues for different types of research. We're not just doing single isolate genome sequencing anymore, but we're doing RNA sequencing, there's ChIP-seq, there's amplicon sequencing, there's a whole slew of different types of goals or applications that we're applying these technologies towards. And now there are centers around the world that are doing exploratory research using genomics and pushing the boundaries of whatever science they're conducting in biology. So with the advent of so many new technologies, especially with big data, comes an influx of algorithms to try and deal with this new type of data. And so this is a nice list compiled by Nuno Fonseca at Sanger, or EBI, listing the growth of number of just alignment tools. These are tools that grab the individual sequence reads coming off your machine and throwing them against a reference or a reference database and seeing what sticks and where. And so you can see that there's a challenge in terms of you picking what tool you might want to use given your particular goal of interest. And so I've been answering this type of question for years whenever I'm being approached by colleagues or friends, you know, what should I use? And I'm glad to have seen that this bioinformatics company in Germany came up with the same answer, that there is no best read aligner. It really just depends on your goals, the type of input sequence, the type of chemistry, and everything else that comes along with science. You have to weigh what tool you're going to use depending on the question you want answered. And so I decided to spend most of my time not making better slides, but researching Google with images. And I thought I'd create this analogy for you. So if you have a single tool, there's a number of things that could go right. There's also a few things that can go wrong. But you need a reference database for your tool. Even if you only have one tool, there's a possibility you're not using it correctly, or at least not as the user, the designer of that algorithm or that tool intended. Perhaps you're using different parameter settings, and that's why your results may vary. Of course, there are different samples that may result in something that's similar but slightly different. Sometimes they're quite obviously different. And then occasionally, depending on how you look at the data, it might look the same as what you've seen previously, but really, if you take a different angle or slice of that data, you might see it differently. And then there's different tools. And the different tools have been primarily designed by grad students to fulfill their thesis project, which is a specific goal with a specific data set with a specific sample type or series of samples. So there's some cautionary use for this. So with all of this complexity of these various technologies, new chemistries that are being thrown at us, all these new applications that we want to use, and various questions, even if you're looking at the same sample, this poses really a pretty grand challenge for most bioinformaticists and most biologists. What should I use and how? And so I tasked this grand challenge to two of my very talented developers to try and solve, and we created this what amounts to a suite of tools or a platform that we call Edge Bioinformatics, which I'm biased. I will try and sell to you, but it's open source. You can look at it yourselves and see whether or not it accommodates what you want to do given your data. So you might also be wondering, why would someone at Los Alamos be playing in genomics? And I posed this question to Brian and Tom earlier today, but they said, of course, Los Alamos has been in genomics for a while. Many of you may not know this. I did not know it before joining the National Lab System. GenBank was initiated at Los Alamos and handed over to NIH about 6 years later. The Human Genome Project really started at the national labs and by the national lab systems. ostensibly to look at radiation effects on the human genome, but they quickly shifted tunes once NIH threw much heavier weight and dollars into the project. And they shifted more towards microbial genomics, metagenomics, fungal genomics, and plant genomics to address other key Department of Energy missions. But we've had a slew of relatively large highlights in the genomics arena at Los Alamos, and our latest attempt is trying to push the boundaries of metagenomic data analysis using a variety of bioinformatics tools, which I'll tell you about soon. So this is a cartoon depicting some of the workflows integrated within our EDGE system. It was recently published last year. It's completely open source, and we were tasked with this challenge to make all these labs that are acquiring sequencers become proficient in genomics or proficient in data analytics. As you might know, if you've ever used a sequencer, it's pretty easy nowadays to generate a lot of data. It's normally much tougher to analyze it, and so that was our task. This is open source. We've made it cloud compatible. We have a VMware image. It's in a Docker container. We have plenty of documentation, and we have 2 web servers. The Los Alamos National Lab site, due to security reasons, prohibits you from updating or inputting any information or analyzing anything new, but this other site is completely open and also harbors additional tools that weren't in the original publication. In essence, the workflow can be relegated to this. All you really need is a FASTQ file or a series of FASTQ files, which is the data that either comes straight off your machine or is considered the raw data from any sequencing machine these days. We have a number of different processes. Some of these require an additional input. For example, if you're choosing to compare your genome to an existing genome, then you obviously need a reference FASTA or a GenBank file or a GFF file. And then we create a number of outputs. I think the trick here is that it's all encompassed within a web-based framework, and you have page projects that display all of your data in the same way that you submit it. Now, I'll run you through some example really quick. We essentially used or recycled a number of pipelines that we had either used for data analytics or that we created ourselves to address certain challenges using new next-gen sequencing technologies. So this is one of the home pages that you see. The only thing required is a project name and for you to point to your data. You can point to an SRA file if it's open to the public and it's in the SRA repository or ENA repository, or you can just point to a mounted drive with all of your data, and you can just select all the data from there. And then you simply need to toggle on or off a variety of different processes. There's documentation behind everything that's being done and all the defaults, and you can modify some of the default settings by clicking on any tab. So this was an example that I ran for live for a conference. I didn't think I'd be able to do a live demo here since I wanted to cover other topics, but you're welcome to go to this site and test it out, or these sites. So if you just plug and play this, it'll also point you to the full-fledged site. Once you set up a run and you selected what modules you wanted to run, it does real-time tracking of the data processing, and it'll tell you what step you're at. It'll also tell you the usage of whatever server that you've opted to choose. You can modify a number of these settings as well, and you can look at a live raw log of all the commands that are being issued. It's also stored in a log file. You can delete projects, share projects, publish them to the open, etc. As you go through, things will keep progressing until it's all done, and then you have these tabs that you can click on. You'll get a summary of how much time anything took and whether or not it completed successfully. You get readouts of whatever step that you used. We provide summaries that we find useful, but if you find something else that's useful, it's actually easy enough to modify this output file so that you can view it regularly. But here we provide a pre and post analysis view. So this is just a QC module where we're trimming and removing poor quality data. And then you can just click through some of the graphs that you might be used to or might not be, but all the explanation is online on how to read some of these graphs to assess the quality of your data and how much data is retained. This is a taxonomy classification module. I'll go over some other examples from other tools, but here we're running a number of different tools. We have more integrated into Edge if you want to use them, but we have limited to some of the ones that are being commonly used out there, as well as to our favorites. We're biased, of course, because we developed some of the algorithms ourselves. But I'll show you the use cases of some of these. But we provide a variety of plots. We're working on making the heat map interactive. We have a radar plot. We provide chronoplots and dendrograms that you might be used to. Everything is toggleable. Also, if you click on the directory, you get access to all the raw data outputs, as well as the converted PNG files. Or we also have access to a genome browser straight from the website, and you can see either the annotation of your contigs or the reference genome and its annotation, and any pileups that you've run using your read mapping or contig mapping to the reference. And you can look at either variants or polymorphisms within the community that you're sequencing or within the isolate that you're sequencing. We've also integrated into this full-fledged phylogenetic workflow. One thing I didn't mention is every single module will accommodate either contigs or reads. And so all the analytics are done two ways to hopefully converge on a single answer, or at least highlight where you might have some issues in analyzing the data or some complexity in your data. So these are just example outputs from the site. And hopefully both your read and your contig analysis come out at the same location for your phylogeny. I thought I'd expand upon this because mostly because I'm trying to pressure my postdoc into writing a paper, which we're almost done. And so he's provided me these slides last night, which I've yet to completely have gone through, but I'm assuming he did a good job. Here we allow any type of input, so reads, contigs, or reference genomes. You need at least one contig set or complete genome set in order to do everything here. So in general, we just find a core genome alignment. We generate a SNP matrix. We infer a phylogeny using either a maximum likelihood or maximum likelihood-like method. And then we also tacked on a molecular evolution package to be able to look at positive or negative selection in one or more genes in your genomes of interest or lineages of interest. And so I thought I'd run you through some of this quickly. We have 5 major steps. Like I said, you need one type of reference. It doesn't need to be a full reference. It can be contigs. We run NucMer, which is a relatively rapid, complete nucleotide alignment. You can also run BWA-MEM. And then you can just find all the sections of these genomes that align to themselves. We have a pre-filtering step to remove all duplicated regions within a genome that might confuse evolutionary inference. And then we map reads onto the reference. We actually have a MASH algorithm to try and choose the best reference to map to first, but then we map that and then we calculate the core. From the core genome, we extract all the columns that contain SNPs, and from this SNP matrix, we infer a tree, and then from this tree and the alignment, we look at positive and negative selection. It also needs an annotation file for that. So we decided to test the general methodology, see if it works en masse. There are a growing number of approaches that try to look at more of a complete picture, looking at whole genome SNPs, but there are few and far between. Very few of them are, I think one of them might have a GUI. Most of them only work on reads. One of them may work on contigs as well, or they only work on contigs or reads. So this one has a lot more full-fledged set of features just to allow a bit of flexibility in your analytics. So this is a tree of E. coli, just recapitulating all the known clades in the right order. The core genome size and other statistics are all available to grab from this. This is an almost invisible tree that also includes a number of genomes from Salmonella, Shigella, and E. coli. The goal of FAME, because it's a nucleotide-based and an alignment-based tree inference, is really meant for fairly closely related genomes. So we wouldn't normally go outside of genus boundary or below 85% identity or so. But we recapitulate all the proper groups. We also discovered a few issues with nomenclature, a few Escherichias that fall outside of Salmonella even. One of them was renamed just a month ago, so we feel that this is on the right track. Also did this, we also did a tree for a very phylogenetically confused genus of Burkholderia. There's a number of different species that within Burkholderia that are very tough to resolve using a number of different methods. And so we want to dig into this. I don't expect any of you to read any of the strain names, but I want to use this one in particular as an example to show that so we grabbed all completed genomes that were published in the RefSeq database. We also grabbed all the initial contig assemblies that we had done within LANL since we generated some of these genomes. We also grabbed all the read data sets that we deposited into SRA, and we threw them all into the same phylogeny just to be able to show you that in green are all the reads, and you can see that they're pair matched with either their contig set or with the completed genome. So we feel that you can do your analytics straight off of the reads. There's no need for assembly ahead of time because we place it in the same location as we would place the assembly otherwise. This is just more Burkholderia. We zoomed into the Burkholderia cepacia complex, which is of interest at least to one of the medical doctors at University of Michigan, John LaPuma, who's done a lot of work in this particular group. And we've identified a few misnamed organisms that fall outside of the entire clade of Cenocepacia. And I'll really only bring this up to highlight the fact that GenBank and RefSeq, as good as they are littered with a variety of different issues. One of them is misnaming. I've found bacterial genomes that are in the viral genome database simply because the strain name matched a bacteriophage name that was oddly named after that strain. Also, I think C. difficile was mentioned during one of my discussions today, and C. diff was changed to P. diff, Peptoclostridium difficile, and of course, because the medical community wouldn't be able to accept not saying C. diff, they rechanged that to Clostridioides difficile. So, so that's an accurate taxonomy label now, but the point is that things can change, and so so many things are very difficult in analysis. For example, if you're using one tool, your favorite tool, but the database was created a day before C. diff changed to P. diff, or maybe the day after. Yeah. And now it's changed back, and you use two different tools, and you're trying to look for confirmation. Obviously, they're not going to confirm the same name because the same name does not exist in both databases. So these are some of the issues that we're trying to tackle as well, but I will go into that a little bit later. We also applied this to Saccharomyces cerevisiae and neighbors, so the entire genus of Saccharomyces, as well as to the recent Ebola crisis genomes that came out. So up to 1,500 genomes, and then all the data sets that are available for Saccharomyces. So we feel like we've spanned the tree of life in terms of having a tool that works on any type of data set and any type of input. Because this is an alignment-based method, we also tested its use in metagenomic samples. So we had a couple of samples that we acquired from CDC, or that we sequenced with CDC from the 2014 German, I shouldn't say German, the E. coli outbreak that happened in Europe. And two Americans came back with symptoms or signs of this 0104 isolate. And so we sequenced these. One of the samples was chock full of E. coli. The other one was moderately, had a moderate amount of data. Over 300 million reads were generated each, so that was a full lane of Illumina at the time. At the time, we assembled the data. We fished out the E. coli contigs, and then we created a phylogeny out of them, and that was published the next year. But here, we just threw all the data as a raw FASTQ file into this tree. So we're searching for E. coli. We know that we want a reference of E. coli, so it shouldn't be that difficult. It's the same way of fishing out contigs. We're just doing it with reads instead. And as you can see here, we clearly get a location of one patient right near this E. coli S88. The other one fits right into the outbreak clade. So this one was positive for that particular strain. This one was not. And on this right side, I just show you a raw read mapping of the data. So if you just took reads and you map them against all genomes and you simply asked, where does this read fit best, map to all possible locations where it fits best, this is the distribution of reads that you'd find for the two samples, patient 1, which is right here, and you see all the data mapping to organisms in this clade. And patient 2 actually had another organism, perhaps a commensal, that was also present in the data set. But because this organism is so much more abundant, the parameters used for doing SNP calling or variant calling with this E. coli in our pipeline place this sample right here, even though it's a metagenomic data set. So as long as you have a clonal isolate that's a dominant in your community, we feel we can process even raw metagenomic data using this tool. So in summary, for the phylogeny part, this is designed for closely related genomes. It's based on whole genome core alignment and whole genome SNPs. It accommodates hundreds to thousands of data sets relatively easily on a traditional server. Can process virtually any type of input. It works across the tree of life, or at least small eukaryotic genomes. We have solutions for larger ones as well. We track coding, non-coding, as long as an input file is given for the reference or for one of the references, so that we can identify synonymous and non-synonymous SNPs. We can identify pretty easily nomenclature issues or issues in some of the deposited isolates, and we're using it now to try and identify incorrectly named organisms throughout NCBI taxonomy. We can process, like I said, metagenomic data, and we output essentially all of the raw output that you might want to use. We do not provide the best trees possible visually, but we're still looking at an array of different visualization methods for phylogenies and even for geographic mapping, which there's very few software items out there. Okay, so this is if you know what you're looking for and you know what you want to do. So you have a target, your favorite organism. You're looking to see where it fits in the tree, or you're looking at a diversity study. You have a number of isolates, or maybe you're looking at epidemiology and you want to track the evolution of an outbreak or something. You can use a phylogenetic approach for that. But for other cases, you may want to simply ask, what is present in my sample? For many environmental studies, this is already done, but they use as a proxy 16S or ribosomal RNA typing. But people are turning to shotgun metagenomic methods to try also to recapitulate what might be in a sample. Another reason to use metagenomics is maybe not in environmental samples, but more for clinical use, trying to identify whether or not a pathogen is present at any burden throughout any stage of the life cycle and the infection, and tissue type. Correct. So 16S would never be able to discriminate. There are many pathogens with very close near neighbors that where you wouldn't be able to discriminate with ribosomal RNA type. So we've included a number of tools, like I showed you earlier. We generate a heat map. We also do a form of contig-based assignment, and we simply categorize them by organism at any level of taxonomy and give you a plot where the contigs that are labeled as a particular taxon get clustered together based on GC and fold coverage, which is what you'd expect for an assembly. So we've revamped the tool at least twice, almost in its entirety, because we had limited scope at first and scope crawl or creep, and that expanded. So things are much more secure. We've added a number of new tools. We have antimicrobial resistance and a virulence database attached to it now that are standardized, and we've had tools to screen both contigs and reads against these two databases. We're creating a full-fledged pipeline for pathogen detection and characterization based on this and the other tools that are already in there, and we're looking at amino acid-based classifiers as well to try and capture what some people call virus hunting, which is to try and discover new viruses that are the cause of some outbreaks. We have an interactive pathway viewer that accommodates a number of different type of omics data. We have a post-assembly binning emphasis to try and bin all the contigs of interest into species-level bins, which is itself a challenge. We have an RNA-seq differential gene expression pipeline, which accommodates metagenomic as well as genomic data, as long as you have a reference or a set of references. We've essentially transformed all the tools that I just mentioned into being able to take long read erroneous data. So we also allow the inclusion of just recently of nanopore generated or Oxford nanopore data into most of these tools, and we're beta testing some of the other tools that are in there, including the phylogeny program. And we're creating a framework and an API for third-party integration of workflows or tools into this system as well. So we've integrated this major effort with two other efforts that we have. One is international engagement. So we have a project where we're engaging a number of different labs across the world to try and get them up to speed on genomics and on the analytics, the bioinformatics. So we've already distributed this package to a number of groups. And from our website, we know it's already been used by about 1,000 different labs. So we feel like we have a number of groups that we might be able to rely on to provide data for biosurveillance. So we have a completely different effort on biosurveillance where we would like to get knowledge as to what background levels of potential pathogens might be in any area. Now, ideally, this would be environmental as well, and we will eventually get there. But there are many farms already that screen animals, and USDA does routine screening of different farms. And then in the clinical setting, one might expect, or it's been proposed numerous times, that sequencing is going to hit the clinic anytime now. We're not too sure where or when that will hit, but at some point there will be sequencing being done relatively routinely for some samples. And so we think this is a great source of information to try and gather. Also, there's tons of data being deposited into the SRA. So we've got about 10 terabytes of data or terabases of data being deposited every month. This is just amplicon and shotgun metagenomic data in blue. And so our goal is to try and funnel data directly. We don't even need to wait for SRA, but we've created a submission tool now as part of Edge where you simply click a button and all the metadata as well as the information of what's identified in your sample can be pushed to a particular server, and then that'll be displayed on a geographic map with a timeline. So in order to do this, you actually need pretty good and reliable taxonomy tools. So you don't want to say that there's Ebola everywhere in the world. That might cause some panic, but you would like to detect it if it's present. So I thought I'd spend the rest of the talk really talking about metagenomic taxonomy classification, which is kind of a very high active area of research and with a number of groups. It's being used by a number of very proficient groups using a number of different tools and examining different types of samples. So like I had said with the hair cutting example, you get what you play with, and so your mileage may vary depending on your goals and the type of tool that you're using. So I thought I'd show you a nice investigation by the Mason Lab looking at metagenomic classifiers. I actually have a lot of critique for all the types of tests that were done because many more could be done, but at some point you have to call it. They investigated maybe 20 or so tools, but as you may be aware, there's tools that are published almost every day, and so this is a year-old slide. Omics Tools doesn't even have this layout anymore. And so there are hundreds of different tools just to do this one task. And so these are the ones that were tested. I'll tell you the GOTTCHA algorithm is ours, and so we are a bit biased, but and I'll let you know a little bit more about how we fare here. This is in terms of greater accuracy. They're looking particularly at this F-score, and there's precision recall here as well in the area under the curve. There's a different metric or an F-score metric for both filtered and unfiltered data as well. In terms of accuracy is up or to the left. We selected tools, so all the tools indicated in red are natively encompassed within our Edge Bioinformatics software, and that's because they run rather quickly. So we did this both for speed as well as accuracy. And in here, we actually chose a slew of different tools, in part because some of them are better for some types of gold or some types of samples than others. And I can get into the details later as well. This is one last chart looking at relative abundance quantification. There is no standard for relative abundance quantification, nor is there a good method to test this yet, at least not holistically. So whatever they tested, this is the order that things came in. I could come up with a series of other data sets where the order would almost be reversed. So why is specificity key? So high accuracy. I thought I'd give this as an example. This is sequencing a Bacillus anthracis strain. So our lab has sequenced a lot of these over the past. So this was one strain that was pooled with a number of other strains in a single lane of Illumina and then sequenced. And you know, the columns are different tools, and you can see that some tools will identify a large number of organisms, even if the tail end is very low or rare biosphere members, let's say. And so the top hits are pretty consistent across the board. We have a couple of viral taxonomy profilers, if you're wondering why those don't line up. But there are a few top ones, so I'll zoom in here. So Bacillus anthracis is indeed the top one, at least in terms of average for all the tools. There are a number of bacillus phages, which also makes sense given the input. And this Staphylococcus phage is also known to be in Bacillus. And then you'll see Bacillus thuringiensis, Bacillus cereus, as well as a few other organisms. So, you know, we were curious why you would see a Yersinia pestis. That makes no sense. Same with Francisella, Filammarasia, or Clostridium. And it's only seen, well, it is seen with a number of the tools. Our tool, which I'll explain to you further how it works, but essentially, if we have a complete genome database, which I realize we don't, the algorithm will not give you a false positive. So also if there's no errors in reads, which we know doesn't happen, but at least with those two caveats, we know our algorithm is extremely specific, but we also identified, we did not identify this Bacillus thuringiensis, which is one of the nearest neighbors to anthracis, which we were happy about, but we did find cereus and all these others, which were confusing until we look at the other samples that were run in the same lane. And if you've heard of barcode crosstalk, you will know that the barcodes that you use to label your samples are not pure barcodes. They're mostly pure. There's only 0.1% contamination frequently, but the tools will pick up those crosstalks of barcodes, other barcodes that are used for other samples that are identified in your sample. Now, this does not matter if you only care about what's abundant in your sample, what's truly relevant in your sample, perhaps. But if you're being sequestered away into one tent if you have Ebola or not, this matters a great deal. And so you'd like to be very accurate in your determination from clinical samples. So I thought I'd run through you. This is a couple. It's in the manufacturer. So we're aware of one company, a spin-off company from our software and our wet lab work that has asked for completely pure barcodes. They also do dual indexing, which helps reduce a great deal. And then they have a couple of additional tricks to reduce this to virtually zero. So there's a number of issues with barcodes, but you know, if you don't care about that 0.01% or you're sequencing an isolate, you know, that contamination will never show up in an assembly and won't really matter in terms of anything that you really do. Yeah, so a number of things that have been carryover, but once Illumina implemented washes between their runs, the only additional lab carryover would be lab contamination or aerosolization stuff, and which still could be, but with some pretty good, robust techniques, you can eliminate that pretty well.
Does it also come from the software, though, specifically from Illumina? So if they have
yeah.
error in one of the barcodes, they don't just discard that. They put it, assign it to the closest barcode.
So you can bypass this. We don't use the Illumina barcoding software, so we try and bypass that. But you can be very, very specific in terms of how you bin your reads into different barcodes. You can do the assignment later.
Right. But if you take the Illumina data that comes
Oh, yeah.
process their way, some of those that barcode contamination created in the software.
Right. Yeah. I can't remember any workarounds to this, but yes, that's a pervasive issue that Illumina is very well aware about. That there's only so much they can do to cater to the 1% or the 0.01% of their industry, of their market. In any case, so I'll just run through you our, in terms of a visual of how we created our database. So the only thing really special about our methodology is the database that we create. And for this, if you imagine for every single organism or every single genome in the database, we identify at the very root of the tree all the external branches. We identify any overlap between all of these genomes and your genome of interest, that single genome of interest. And so we identify these locations, we remove them from the database, or actually we simply notate their coordinates and put them aside. We do this for every level of taxonomy up to the strain level. And what remains is the unique fraction that's completely unique to that strain, given everything else in the database. So this reduces the complexity of your database by a large amount. And so now we can use essentially any tool to map these reads back to these leftover fragments to see where they might belong and to which genome they might belong at the strain level. We can do this at every level of taxonomy, actually, so that you can still look at the core that's conserved within all of E. coli, for example. So, so this is the setup. There's a number of reference unique segments that remain at any level of taxonomy that you care about, and you can count the number of reads that map onto these data. Most of the tools out there will simply give you read count as abundance. If you're comparing a virus with a bacterium or with a eukaryote, maybe read count would not be the best metric for relative abundance in your sample. So we've come up with a couple of additional metrics, which is the % of the linear coverage of your unique segment that you're covering, as well as the fold coverage within that covered segment. And it turns out that these two metrics can really aid in your relative abundance calculation. This is presence-absence only, however. We did not imply, or we did not put any cutoffs on any of the tools. These are all defaults outside of BWA and BLAST, where we used a N of 5 or 10 cutoff. But here's the results. So you can see that some of the tools have an extreme false positive rate. This is an artificial community that was actually sequenced. So it's physical strains that were mixed together, sequenced on Illumina. So we know the composition, and these are the results. Anything negative was actually just missing from the databases. So they're actually very good at finding whatever's present in their databases, but some of them are less good at telling you only what's present in your sample. So we use this, and this was a couple of years back, though. We've been working on it since, but there's a limitation to the method that we just created, which is, despite going all the way up to kingdom level, we have all of these databases that we need to probe to do this, and actually calculating this database is somewhat difficult, although we're really trying to work around it. So we thought of a regular approach, but based on read mapping, so that you did not need to, you could create databases a little bit easier on the fly, instead of trying to pre-compute an index or a database and then have to map against it. But from what I understand, there's maybe a method developed here at the University of Michigan, which I'm anxious to try for mapping as well. In any case, so we've learned a lot from our efforts. We have collated a pathogen database. Oddly enough, no one has a complete pathogen list, not even CDC or WHO. So we've collated our own, and we were trying to automate that process so we don't have to do it again next year. We have our genome uniqueness database, which comes from the previous tool I just went over. You can enter in any host. So, you know, we may be concerned about ourselves, but some people are concerned about their cattle or other organisms. We have a particular control file. We can add a custom strain taxonomy, since we realize there's a very big difference between our idea of taxonomy and actual phylogenetic evolution. And metadata for all the pathogens that we've described in our pathogen database. We still look at all the levels of taxonomy, similar to a last common ancestor or lowest common ancestor algorithm. We also do a rank-specific mapping based on what rank they are most similar to, which I'll get to in a minute. And we also tried to put in a scoring method that was less or not reliant on abundance. So if you look at any type of tool, what makes you confident that something's present in your sample? It's natural to think that the more data you have, the more confident you are. And that's great, unless you're looking at low abundance pathogens in a sample. You would like to be very confident that a pathogen's in your sample despite the abundance. Or if you expect that something should not be very prevalent, but you should expect it present, you'd like to have it there. Yeah, I should check what time. Okay, so I'll run through this relatively quickly. I apologize. So this is like an LCA method. We map data to all of these genomes, and I'm just giving you a rote example, a made-up example of a read that may map to all three of these strains, a read that may map only to this E. coli strain, and a read that maps to this E. coli strain and this Bacillus ames strain. And this is the matrix that we work off of. And then we normalize the data by the number of references that every read hits. We normalize by identity, so most of the tools don't accommodate % identity of their actual map. They just call a presence or absence. And we can normalize by both. How much depth is enough? Because we have the uniques information, we have maybe some idea of priors as to what will be mapping to signature regions. And so we have some expectation as to the distribution of the data along the genome. And so we estimate the probability of this genome given the distribution of the reads at every level of taxonomy. And we know that every genome has different fractions of it that are unique at a different level of taxonomy. And so we essentially take the inverse of this and calculate a probability distribution from this. And we can come up with a score on what's the expectation of finding, a distribution that we see throughout any given genome in our database. And we've tested this with over 700 contrived samples that were spiked with 4 different types of pathogens at different levels. We've also tested it on hundreds of different samples that are synthetically generated. It appears to have a fairly decent balance between sensitivity and specificity. I didn't mention the other scoring function, which is to give it a negative control. So if you have a healthy sample from day 1 and you're sick day 2, you could compare to yourself. In any case, I thought I'd just run you through our idea of challenges. In terms of taxonomy classification, once you have the tool, you think it might be easy. It's really not. And that's mostly because we neglect this entire side of the conversation, the database side. And the databases are growing. That's why this prediction was out there that genomics will surpass all other big data analytics combined. Even NCBI has stopped the use of being able to explore all of WGS using BLAST because it's simply too large and too resource intensive. There are a couple of publications on problems with too much data, and I just thought I'd run you through the different databases. So when I say NCBI or RefSeq and you say NCBI or RefSeq, we may be talking slightly different languages. This is the actual distribution of data in the BLAST-NT database. This is RefSeq. This is GenBank in blue. This is the WGS. WGS does not overlap at all with GenBank, just in case you were wondering. RefSeq has many genomes in the GenBank, in BLAST-NT, but also outside in the WGS, in the whole genome shotgun archive. Bacteria here outnumber and outpace even the BLAST-NT database. And oddly, interestingly, bacteria, archaea, and viruses are a very small fraction of this because there are a lot of eukaryotic, particularly human reference genomes in here. But most tools really only look at a subset of this information. They do not look at all of this. So we've revamped our GOTTCHA 2 algorithm. We had to stop using a variety of different languages and turn straight to C++ and a number of computational science tricks to be able to do this. And we now have a database for all of RefSeq, including the eukaryotes. Of course, that was last year, and so this year things have changed. some of the databases have essentially doubled in size. And so this is a constant growing challenge. Our current estimate for database construction will be about 500,000 CPU hours. And we need at least 50 terabytes of space just to be able to construct the database. So this is a grand challenge. I only talked about bioinformatics. We have a number of individual funded projects with other companies, our spin-off companies. We look at other technologies. We have natural product research goals, interactions or soil metagenomics, phylosphere or plant metagenomics, human metagenomics, and a number of other things. And obviously, this is not me and my group alone. My group is fantastic. I have a number of great postdocs and bioinformaticians. We work very closely hand in hand with our wet lab genomics counterparts and our microbiology group. With that, I'll take any questions. This is just a video of me live doing my demo at a different time. I can't remember when. Thanks.
Lecture's recorded, so we'll try to capture your questions with a microphone on that side and this side. While you're thinking of the questions, hey, Patrick, one of the views that you gave in the metagenomes was with the pathway viewer. What metabolic potential was there? Of course, I mean, lots of us are measuring the metabolites too. Are you giving thought about how you're going to handle getting the metabolite data integrated with that?
Yeah, that's a great question. We've thought about it. We haven't come to a conclusion. So adding transcriptomic data is easy. Proteomic data, pretty easy to that omics viewer map. Adding the metabolite data, I think metabolomics is that cutting edge field that's very tricky, particularly for metagenomes, particularly for non-typical metagenomes. Let's consider human microbiome as a normal environment that metabolomics is being applied to. So we know a lot more of the metabolites there. We can identify a lot more of the peaks in terms of actual metabolites, and so we can place them on a map. But in other environments, most of the peaks are left uncharacterized. You have no clue what metabolites they are, and how do you still track those and try to display them? They can't be displayed on the map very easily. So I'm not sure. I'd welcome any ideas. We are not. Our focus has been genomics primarily, as you've seen. We're starting to play with other types of data sets outside of sequence-derived data, but I'd be welcome to collaborating with anyone who has great ideas on how to visualize, how to transform the data, and really push the boundaries on what's possible given today's work. And we have a couple of interactions with PNNL that have a very large mass spec facility, and they're experts both in metabolomics as well as proteomics, and we're trying to work with them to try and explore what's possible in terms of data analytics.
It's a really cool set of work. So when you were showing all the plots like this, right? Sorry, it just went down. Plots where you're comparing performance for several different tools for the identification of species that are present. How much can you gain if you just take a meta prediction or a consensus across all these different tools that exist?
Yeah, so that's almost what the heat map does. It kind of rank orders based on the average for all within tool normalized relative abundance criteria. Our personal preference, I mentioned my bias considering we developed our tool, is to turn to our GOTTCHA analytical tool for what's really present. Virtually guaranteed to be there, if not the nearest neighbor to that thing, and nothing else closer to it is available in the database. So that's our first go-to. If we're more in an exploratory mode for an organism that we think should be there, but we don't find it in our list, then we go down to a different tool that might be a bit more lenient in being able to identify organisms. So We turn to our Pangea tool now. We've turned to Kraken, to BWA. Different methods might be appropriate.
But can you take like a formal consensus and look at how it performs on those metrics you were looking at?
What do you mean?
Well, so just let all the methods vote, for example, about whether or
Right.
not a given thing is present, right? And then how does that perform on your standard statistics compared with any of the individual models? It should be better. I'm sort of wondering how much better, and especially can you push the boundaries on either the sensitivity or specificity?
I've had this conversation with other groups that are, I think some other groups are trying to do that. We have not looked into it. In part, I mentioned this taxonomy issue. That happens to be quite frequent, not because of changes in taxonomy, but more because of when the databases were constructed. Some of the tools that are prevalently used in some facets of metagenome research or microbiome research, like Metaflan, you cannot create a new database. You just have to wait for one. Therefore, unless you know what's in the database, it's very difficult to ascertain why you may or may not find a particular organism in there. So we had not really considered. I think that would be appropriate, especially if you normalized all the databases. But we've turned to trying to optimize one or two tools to be able to capture the diversity, but still be very specific, still be pretty accurate in your calls, to try and limit the number of false positives. But looking at consensus, yeah, maybe I'll ask. I can task someone with that, as I do nothing myself anymore.
So I feel like in general, especially when we're trying to validate these tools that are supposed to go down to the strain level, frequently people are using artificial communities that never have strain level conflict. And you mentioned that that actually might be a problem also in this case for your tool. Can you talk a little bit about if you were able to validate in cases where you have close relatives in the same sample, and then also what you expect to happen using this algorithm when that occurs?
Yeah, sure. So first, the algorithm question. With our GOTTCHA algorithm, we look for perfect k-mers to remove sections that are identical. But when we do the search, we accommodate and tolerate a few additional mutations to accommodate possible evolution or diversity of strains. We actually use a larger k-mer than was used to remove similar k-mers. So we use, I think, like a 24-mer or 26-mer for removal. We use a 30-mer for actual mapping. But we accommodate 2 nucleotide variabilities. That's default settings. You can augment that. We have tested how that works in real situations to identify new strains. We feel that that does an okay job of identifying. We haven't tested what the limits are. You know, if you only have 10 reads in a sample that map, you may get rid of a few additional reads. And so what is your readout at that point? There's a big effort on strain challenge that's being run right now by, I believe it's maybe FDA. It's hosted by DNA Nexus, I believe. And so that challenge is supposed to get to, oh no, it's a Johnson & Johnson or a Johnson & Johnson derivative that's looking into this. And the goal there is to look for what kind of tools and utilities are really good at strain-to-strain discrimination. I don't know if a taxonomy classifier should be truly used down to the strain level. For strain level, it's far better to have actual alignments with sequences and maybe use a phylogenetic approach instead to really place or characterize some of these sequences. But there is an effort, and some of them use ideas like phylogenetic approach to try and deconvolute strains. That said, we still have used our GOTTCHA tool, and as long as it is one of the strains that's in our database, you can actually place it quite specifically. Given enough data, should caveat everything.
Patrick, thanks a lot. glad to put you face to face with some people, and hope you see you around campus more. Yeah. Join me in thanking Patrick again.
So since we stopped recording, I can also say synthetic data